Good day everyone. Today I will be discuss about DNA model, replication, and transcription. First, let's discuss what is DNA. DNA or deoxyribonucleic acid is a molecule composed of two polynucleotide chains that coil around each other to form a double helix carrying genetic instruction for the development, functioning, growth, and reproduction of all known organisms and many viruses. By making a wire model of the structure, it became clear that the only way to model could conform the requirements of the molecular dimensions of DNA was if A always paired with T and G with C. In fact, the A and T and G and C pair showed a satisfying lock and K fit. DNA replication is one of the most basic processes that occurs within a cell. Each time a cell divides, the two resulting daughter cells must contain exactly the same genetic information or DNA as the parent cell. Replication occurs in three major steps: the opening of the double helix and separation of the DNA strands, the priming of the template strand, and the assembly of the new DNA segment. During separation, the two strands of the DNA double helix uncoil at a specific location called the origin. Several enzymes and proteins then work together to prepare or prime the strand for duplication. Finally, a special enzyme called DNA polymerase organizes the assembly of the new DNA strand. The following description of these three stages processes applies generally to all cells, but specifically, variations with the processes may occur depending on organism and cell type. Transcription is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA. DNA safely and stably stores genetic material in the nuclei of the cell as a reference or template. Transcription is carried out by an enzyme called RNA polymerase and a number of accessory proteins called transcription factor. Transcription factor can bind to specific DNA sequences called enhancer and promoter sequences in order to recruit RNA polymerase to an appropriate transcription site. Together, the transcription factors and RNA polymerase form a complex called the transcription initiation complex. This complex initiates transcription and the RNA polymerase begins mRNA synthesis by matching complementary bases to the original DNA strand.